Hello friends, I am MD Harun Al Rashid and it's my YouTube channel Harun by John. In this video, I'll fully explain the pentose phosphate pathway or phosphogluconate pathway or hexose monophosphate pathway. This pathway is very important for different higher level competitive exams. Okay, so don't be late. Let's start. First, what is pentose phosphate pathway? Pentose phosphate pathway is a metabolic pathway that is alternative of glycolysis and Krebs cycle. By this pathway, the glucose oxidized to produce pentose, sugar like ribose and mainly NADPH. Okay, why this process is very important? Because say for example in RBC there is no mitochondria, but here by hexose monophosphate shunt pathway the RBC produce NADPH by this mechanism or by this oxidation pathway. The breakdown of carbohydrate takes place in the body by glycolysis. Normally আমাদের শরীরে carbohydrate breakdown occur by glycolysis and tricarboxylic acid cycle and produce ATP. Okay, the oxidation pathway in our body mainly creates a glycolysis and Krebs cycle. Okay, by this process we generate ATP. The body generates ATP. Okay. But there is another alternative pathway is called pentose phosphate pathway. Glucose oxidation process also occur by pentose phosphate pathway. Okay, also called hexose monophosphate shunt. The main purpose of this pathway is to produce NADPH and pentose sugar and also others derivative like glycerol 3-phosphate, erythroid 4-phosphate, fructose 6-phosphate, etc. that use use in glycolysis. The fate of glucose whether undergo glycolysis or hexose monophosphate pathway is decided by the relative concentration of NADP plus and NADPH. So this type of glucose is enter either glycolysis or PPP or pentose phosphate pathway. What is the cue of this reaction? In the blood, when in the blood the concentration of NADP plus is to NADPH. Concentration is high in when NAD plus concentration is high in blood, then the oxidation reaction occur by pentose phosphate pathway. When the NAD plus pH concentration high in blood, then glycolysis or Krebs cycle occur. This pathway is occur in adipose tissue, that is fat tissue, liver, erythrocyte, testes, adrenal glands, lactating mammary glands. In this place, this pathway is occur. The main purpose of this pathway is oxidation of glucose. It is play primary role in anabolic rather than catabolic. But glycolysis is a anabolic and catabolic both process. Okay, that is amphibolic pathway. Glycolysis Krebs cycle is also called amphibolic pathway, but this is anabolic process. যেখানে তোমার সুজীবদের শুষ্কজনটা বৃদ্ধি পায়. It is important pathway that generate precursor of nucleotide synthesis. তো এখানে যে পেন্টোজ সুগারগুলো ফর্মেশন হলে পেন্টোজ সুগারগুলোর কাজ হচ্ছে তোমার সেখানে নিউক্লিওটাইড তৈরি করা. কারণ নিউক্লিওটাইডের কম্পোনেন্ট অফ নিউক্লিওটাইড ইজ রাইবোজ সুগার, phosphate group and nitrogen base. This pentose sugar is used to produce nucleotide. Okay, and is very important in red blood cells. Why? Because in red blood cell there is no mitochondria, so no Krebs cycle occur in red blood cell. but red blood cell require NADPH. Okay, this NADPH in red blood cell produced by this pentose phosphate pathway or hexose monophosphate shunt. Clear? The PPP is necessary to support nucleotide synthesis. The pentose phosphate pathway purpose কি হচ্ছে? First, nucleotide synthesis, not directly nucleotide synthesis. The component of nucleotide ribose 5-phosphate is produced by pentose phosphate pathway. Preserve cellular redox balance. Cellular redox balance maintain করা আর guard against oxidative stress. আমাদের metabolic processের মাধ্যমে যে সমস্ত অক্সিজেন, হাইড্রোজেন পারঅক্সাইড, সুপার অক্সাইড তৈরি হয় সেগুলোকে গুড ম্যাটেরিয়ালে কনভার্ট করে দেওয়া হচ্ছে এর কাজ। অর্থাৎ আমাদের বডিতে যে হাইড্রোজেন পারঅক্সাইড তৈরি হচ্ছে সেটা কনভার্ট করে দেওয়া H2O এবং O2 তে। এটা হচ্ছে আমাদের অক্সিডেটিভ স্ট্রেস থেকে সেলকে প্রোটেক্ট করা। The hexose monophosphate shunt is occur by two steps. Number one is the oxidative steps where the oxidation reaction occur and non-oxidative phases. Okay, the overall reaction of this pathway is where the glucose 6-phosphate molecule oxidize in the presence of NADP plus and produce ribose 5-phosphate and NADPH and H plus. This NADPH and H plus is required to protect cell from oxidative damage. And this ribose 5-phosphate is required to produce nucleotide. That is the raw material of DNA and RNA. The first step is oxidative pathway. The oxidative phase of this cycle is occur in the cytoplasm of most of the cell, and is regulated by the enzyme glucose 6-phosphate dehydrogenase. This enzyme glucose 6-phosphate dehydrogenase is the main regulatory component of this pathway, of this cycle, and also it is maintained by the concentration of NADP plus that is located in the blood. Okay, and the level of reactive oxygen species, because to protect the cell from oxidative stress, the NADP plus is required to reduce the glutathione component and break down the reactive oxygen species into simpler form. Okay, so এই যে রিয়াকশনটা ডিপেন্ড করছে কার উপরে? গ্লুকোজ 6-ফসফেট ডিহাইড্রোজেনেজ অ্যান্ড NADP প্লাস কনসেন্ট্রেশনের উপরে। প্রসেস এবার পারপাস কি? অক্সিডেটিভ ফেজের পারপাসই হচ্ছে তোমার NADPH তৈরি করা। NADP নিকোটিনামাইড, অ্যাডেনাইড, ডাইনিউক্লিওটাইড, ফসফেট অ্যান্ড হাইড্রাইড, এটা আমাদের তৈরি করতে হবে। The process key, glucose 6-phosphate is irreversibly oxidized to ribose 5-phosphate. The purpose of this process where the glucose 6-phosphate is convert to ribulose 5-phosphate, okay, and generate NADP plus pH plus H plus. The key enzyme, first enzyme is glucose 6-phosphate dehydrogenase that oxidize glucose 6-phosphate, that is aldose sugar, into the ketose sugar, that is 6-phosphogluconolactone. 6 carbon containing glucose 6-phosphate converted into 6 carbon containing ketone sugar, 6-phosphogluconolactone. Okay, in the presence of then 6-phosphogluconolactonase. This 6-phosphogluconolactonase in the presence of magnesium ion and H2O is converted into another aldose sugar, 6-phosphogluconate. Okay, this 6-phosphogluconate in the presence of NADP plus and 6-phosphogluconate dehydrogenase enzyme, that is irreversible reaction, produce the ketose sugar ribulose phosphate, ribulose 5-phosphate. Okay, this ribulose 5-phosphate in the presence of phosphopentose isomerase produce ribose 5-phosphate. তো এখানে কি হলো, গ্লুকোজ 6 ফসফেট ডিহাইড্রোজেনেজ এনজাইমের প্রেজেন্সে যেখানে এনএডিপি প্লাসটা কি হলো অক্সিডাইজ হয়ে গেল, রিডিউস হয়ে গেল, এনএডিপি এইচ প্লাসে প্রডিউস করলো কিটোজ সুগার 6-ফসফোগ্লুকোনো ডেল্টা ল্যাকটান ইন দ্য প্রেজেন্স অফ 6-ফসফোগ্লুকোনো ল্যাকটানেজ এনজাইম. In the presence of magnesium ion and water, it converts into the aldose sugar 6-phosphogluconate. Again 6-phosphogluconate in the presence of 6-phosphogluconate dehydrogenase enzyme produce ribulose 5-phosphate. Okay, this ribulose 5-phosphate is the precursor of nucleotide synthesis. The ribulose 5-phosphate in the presence of isomerase produce D-ribose 5-phosphate. The main purpose of this enzyme is produce NADPH. 2 NADPH are produced in this reaction. Then number 2 is non-oxidative phase. In non-oxidative phase, different sugar phosphate are converted into one another. In this, glyceraldehyde 3-phosphate produce, gyrulose 5-phosphate produce, and fructose 6-phosphate produce, erythrose 4-phosphate produce. Different type of sugar are interconverted and produce, that is again used in the glycolysis pathway. Purpose to interconvert sugar phosphate producing ribose 5-phosphate and other intermediate that can enter glycolysis. There are different type of sugar are produced that is used in different purposes in cell. Clear? The process and key enzyme. The first, the D-ribose 5-phosphate that is produced by oxidative phase, that is D-ribose 5-phosphate. Then in the presence of isomerase enzyme, that is reversible reaction, produce epimerase enzyme produce gyrulose 5-phosphate. Gyrulose 5-phosphate is 5-carbon containing sugar, ketone sugar, and ribose 5-phosphate is also 5-carbon containing aldose sugar. This is aldose and this is ketose sugar. Total 10 carbon is produced. This 10 carbon is converted into pseudoheptulose 7-phosphate. This is a ketose sugar that contains 7 carbon, and glyceraldehyde 3-phosphate, that is aldose sugar, contains 3 carbon. So this total 10 carbon, again in the presence of a transaldolase enzyme, they produce the ketose sugar fructose 6-phosphate, ketose sugar, that is 6 carbon containing, and 4 carbon containing erythrose 4-phosphate is aldose sugar. In both reaction, ketose and aldose, both type of sugar are produced. Okay, this erythrose 4-phosphate again join with gyrulose 5-phosphate, 4 carbon, and gyrulose 5-phosphate is 5 carbon to produce fructose 6 sugar, fructose 6 carbon containing and 3 carbon containing glyceraldehyde 3-phosphate. Clear? And again the pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate produce 6 carbon containing ketose sugar and H4-phosphate. This fructose 6 carbon containing fructose sugar in the presence of isomerase enzyme produce aldose sugar, glucose 6-phosphate, and the fructose 6-phosphate, phosphohexose isomerase enzyme in the presence of isomerase enzyme produce glucose 6-phosphate. Glucose 6-phosphate then again enters into the glycolysis pathway. Clear? The end product formed are used up the other metabolic pathway like glycolysis and Krebs cycle to form glucose. The ultimate glucose 6-phosphate to convert into glucose and enter into the glycolysis. So why these several steps are important? Because by pentose phosphate pathway, several compounds like erythrose 4 phosphate, fructose 6 phosphate, glyceraldehyde 3 phosphate, or ribose 5 phosphate, etc. Ribose 5 phosphate, etc. are produced. This is the derivative of another metabolic component. Significance of HMP shunt. The first significance is produce NADPH. NADPH. Because NADPH is a reductant that reduce and convert and protect cell from oxidative damage. Protect cell from oxidative damage. Clear? The first, in the glucose 6 phosphate to 6 phosphogluconate produce in the presence of glucose 6 phosphate dehydrogenase produce NADPH. Okay, NADPH plus is reduced to produce NADPH plus H plus. This NADPH H plus in the presence of glutathione reductase converted into oxidized glutathione is reduced to glutathione When in the presence of glutathione peroxidase, the reduced glutathione is oxidized to glutathione, and the hydrogen peroxide that produced by metabolic pathway, different metabolic pathway or oxidation reaction. This oxygen is converted into superoxide that joined with proton and produce hydrogen peroxide. Hydrogen peroxide is highly toxic for our cell that cause oxidative damage of cell. This reduced glutathione in the presence of glutathione peroxidase converted hydrogen peroxide into H2O and O2, water. In another reaction, that is in the presence of catalase enzyme, this NADPH2 is required to convert hydrogen peroxide to H2O2. So when আমাদের শরীরে যখন hydrogen peroxide formation হচ্ছে, that is highly toxic to our body, this hydrogen peroxide is in the presence of some NADPH H plus that produced by pentose phosphate pathway converted into H2O and water. So our cells is protecting from oxidative damage. Number 2 significance, the pathway produce pentose sugar, that is ribose 5 phosphate. This ribose 5 phosphate is the raw material of nucleotide synthesis. This is the ribose 5 phosphate. Okay. Number 3, it play an important role in CO2 fixation. Okay, CO2 fixation because through a ribose 5 phosphate. Why? Ribose 5 phosphate derived from ribose 5 phosphate. Ribose 5 phosphate is the ribose 5 phosphate is derived. Ribose 1,5 bisphosphate is derived from ribose 1,5 phosphate. Okay, ribose 5 phosphate is 1,5 bisphosphate that is in the presence of Rubisco enzyme. Rubisco enzyme, ribose bisphosphate, carboxylase oxidase enzymes, ribose 1,5 bisphosphate act as a carboxylation process, carbon dioxide absorb. during photosynthesis. And this ribulose 1,5-bisphosphate is produced from ribulose 5-phosphate. Ribulose 5-phosphate is the product of pentose phosphate pathway. Okay. Number 4, this pathway is involved in the production of several intermediate compounds like ribose 5-phosphate, glycerol 3-phosphate, erythrose 4-phosphate. These are glycerol 3-phosphate, erythrose 4-phosphate, fructose 6-phosphate, cetacrose 7-phosphate, and ribose 5-phosphate. All are essential for the synthesis of nucleotide and other important biomolecules in the cell. The HMP shunt is interconnected with other metabolic pathway like glycolysis in this because এই প্রসেসের মাধ্যমে যে গ্লুকোজটা ফরমেশন হলো, যেখানে আমাদের গ্লুকোজ মানে মেইনলি এই ফ্রুক্টোজ 6-ফসফেট থেকে কি করে ফ্রুক্টোজ 6-ফসফেট থেকে আলটিমেট আমাদের আবার গ্লুকোজ 6-ফসফেট ফরমেশন হচ্ছে, যেটা ফসফেটেজ এনজাইমের মাধ্যমে আবার কনভার্ট হচ্ছে কিসে গ্লুকোজে কনভার্ট হয়ে যাচ্ছে। সেই গ্লুকোজটা অ্যাগেন ইউজ in mitochondria containing cell or cells, cytoplasm and mitochondria to produce energy or ATP. The regulation of pentose phosphate pathway. As you know, the pentose phosphate pathway is highly regulated by glycerol glucose 6-phosphate dehydrogenase enzyme, and the because and concentration of NADPH in cell. Okay, NADPH product act as feedback inhibitor of glucose 6-phosphate dehydrogenase. Okay, this enzyme activate when NADPH level become low in blood or in cell. Okay, so NADPH is a negative feedback inhibitor, and also in the high concentration of ATP in blood inhibit the glucose 6-phosphate dehydrogenase. The ratio of NADP plus and NADPH in the cell also affects PPP regulation. High NADPH level inhibit glucose 6-phosphate dehydrogenase. When NADPH level is high, the glucose 6-phosphate dehydrogenase enzyme inactivate. When NADPH level is low in blood, the glucose 6-phosphate dehydrogenase enzyme activate to produce more NADPH. Hormonally, insulin and glucagon also regulate this pathway. How? Because insulin stimulates glucose absorption into the cell. Okay. When glucose concentration in blood is high, the insulin remove the blood glucose into the cell. Clear? The availability of substrate for then this glucose again converted into glucose 6-phosphate or other molecules by pentose phosphate pathway. But on the glucagon, on the other hand, inhibits the PPP by reducing insulin level. Okay, and glucagon convert glycogen into glucose and increase the concentration of glucose in blood. But insulin, the blood glucose converted to glycogen or transfer into the cell. Okay, the insulin stimulate the PPP pathway, but glucagon inhibit it. Pentose phosphate pathway also controlled by genes like transcriptional factor like NRF2, that is nuclear factor erythroid 2 related factor 2. Okay, this gene trigger in response to cellular stress when in our body or in our cell toxic superoxide, hydrogen peroxide, etc. are produced. That is, cellular stress level is high, then reduce that stress, the transcriptional factor NRF2 activate the pentose phosphate pathway, because NADPH is required to destroy this superoxide or hydrogen peroxide molecules. High quantity of glucose 6-phosphate can stimulate. When the concentration of glucose 6-phosphate is high, then also stimulate this process. So, regulatory substance glucose 6-phosphate concentration, NADPH concentration, insulin and glucagon level, and several stress level, etc. control this pathway. I hope your concept is totally clear about this pathway. If you like it, please like and subscribe my channel. Thank you for watching this video.